So let's do objective 4 is really differentiating between gray matter and white matter and identifying how the myelin sheath is different in the CNS and VNS. Okay. So in the central nervous system, the myelin is created by oligodendrocytes. We talked about that already. And we also even mentioned that an attack on the oligodendrocytes gives us what disease? What disease is created by an attack on oligodendrocytes? MS, good. Multiple sclerosis, that autoimmune attack of these cells that disrupt the myelin. So one oligodendrocyte basically extends to several axons and creates that myelin sheath in that way. So I'll point it up here. So this would be an oligodendrocyte. And as we can see, it's projecting to many different neurons here, or many different axons, and insulating or myelinating these neurons. Now this is only found in the central nervous system, oligodendrocytes. It produces the myelin sheath, but not the neurolemma. We're going to talk about what the neurolemma is in a little bit. Okay, it gives myelin myelin has a white appearance because of the fat component of myelin. It appears white, so we call that white matter. Whereas gray matter is the cell bodies, the dendrites. They lack myelin, and we call that gray matter because it's not myelinated. Right? So if you think about this in this way, if we look at a slice of brain tissue, we're going to see some areas being gray and some areas being white. Right? So such as this image. The gray areas are the areas that are non-myelinated. This is just your exposed bare neuron. That's either a cell body or let's say the dendrite. It does not have myelin wrapped around it. That's your gray matter. The white matter, which is sort of more centrally located here, is white in appearance because it is myelinated, and myelin is really fat. And so it appears more white when we look at this, when we stain this tissue, and that's why we call it white matter. And this is really the myelin that's appearing white. So that's the difference between gray matter, non-myelinated nerves, essentially, and white matter, myelinated nerves. Now, the neurolemma and myelin. Neurolemma is essentially the structure that is created when Schwann cells wrap around the axon. This is in the peripheral nervous system. So what we were speaking about with oligodendrocytes is exclusive to the central nervous system. When we cross to the peripheral nervous system, these are your peripheral nerves, your cranial nerves, your motor neurons. These have what's called neurolemma, which is the Schwann cells that wrap themselves around the axon of the neurons. Okay. So for example, this would be a neuron in the peripheral nervous system. This is its cell body, this is its axon. Now, myelin is going to wrap around the neuron. And again, there are some gaps which we call, what do we call those gaps in myelin? What do we call those gaps in the myelination of a neuron? Nodes of von Baer. Good. So these are the unmyelinated bits that are down the length of this axon. And then the myelinated stretches here are what we call neurolemma. And these are really the Schwann cell that is wrapping around the neuron and myelinating it. Okay. So a lot of students think that Schwann cells make myelin. That is not true. Schwann cells themselves act as the myelin. So they're sitting around the neuron and they myelinate, or they hug that neuron and create neurolemma, which is the myelin sheath. So these wrap around the axon and form this myelin sheath. And again, just to reiterate here, the areas where there's no myelin are called the nodes of von Baer. And these are intentionally left open to speed up the conduction velocity down the length of that neuron. Alrighty, so two takeaway points here. Neurolemma, which is specific to the peripheral nervous system, is really formed by the Schwann cells as they tightly hug the axons. Okay. The second takeaway point here is that there are some intentional gaps in between those Schwann cells that are the nodes of Ranvier that speed up the saltatory conduction down the length of that neuron. Okay. So to talk more about myelin in the peripheral nervous system, there are some small axons, very tiny axons that are usually not even myelinated. Okay, these are going to obviously conduct impulses much slower than the larger. Okay, but they also will have some neurolemma associated with them, so they will still have a Schwann cell with a plasma membrane, but they are unmyelinated. All right, so this would be your myelinated axon here. This is your neuron, essentially, the cell membrane of your neuron. This is your cytoplasm inside. This is the Schwann cell, which is closely adhered to the neuron, sort of hugging the neuron. That's the way I think of it. Okay, and it's in hugging the neuron, it creates this insulation, which we call myelin. Now, if you think about an unmyelinated axon, which is here, we can see that again, it's much smaller and it doesn't have that Schwann cell tightly adhered to it. And obviously, it's going to conduct impulses much slower than that larger myelinated neuron. Okay, so myelin insulates a neuron and speeds up the conduction velocity. All right, any questions so far? Any questions? Sure, so myelin insulates the neuron and speeds up the conduction velocity, how fast that action potential is propagated down the neuron. So a bigger neuron, which is myelinated, will conduct action potential impulses more rapidly than a smaller neuron, which is most likely unmyelinated.